videos report a novel gene transfer technique for differentiation of osteoclasts in vivo. We also describe cell culture protocols for differentiating mouse's bone marrow macrophages of human PBMCs into osteoclasts. This video will be broken into 3 parts. 1. Hydrodynamic delivery of rank ligand mini-circle DNA. 2. In vitro osteoclast generation from mouse bone marrow macrophages. And 3. In vitro osteoclast generation from human PBMCs. Part 1. Hydrodynamic delivery of rank ligand mini-circle DNA. The following animation shows a brief overview of hydrodynamic delivery via mouse tail vein injection. First, place mouse under a heat lamp for 10 minutes prior to injection. This dilates the blood vessels and makes the lateral veins more visible. Then, locate the injection site and proceed with the injection, making sure to complete the injection within 5 to 7 seconds. The next portion of this video will show a more detailed account to the procedures involved in the hydrodynamic delivery via mouse tail vein. Gather the materials for the injection. Warm up the mouse in a cage for 10 minutes prior to injection. Monitor mouse carefully to avoid dehydration and hypothermia. Warming the mouse dilates the blood vessels and makes the lateral veins more visible. Once the veins are visible, transfer the mouse to a restrainer and insert a plunger to restrain mouse movement. Disinfect the injection site with an alcohol wipe. Using a 27 to 30 gauge needle for the mouse tail injection with diluted rank ligand.